Hi, hello, dear students. This is your Harikrishna sir, and today we are going to see the link reaction. Already we have gone through the basic information and first stage of aerobic respiration, that is glycolysis. In the last class we have seen glycolytic pathway. So today we are going to see the link reaction, which is considered as the second step of aerobic respiration. Okay, so since there are four steps or four stages in aerobic respiration, the first one is glycolysis, the second one is link reaction, the third one is Krebs cycle, and finally we have got ETS, electron transport system. Okay, so now what is meant by link reaction? Why it is link reaction? Because of it links or it connects glycolysis and the Krebs cycle. Okay, it links or it connects two major pathways, that is glycolytic pathway as well as the Krebs cycle pathway. So it connects link. It acts as a link for both the metabolic activities. Clear? Hence it is called as link reaction. So two kinds of reactions taken place in this particular reaction. One is what? Decarboxylation. Okay. Then the second one is oxidation. Okay. So two kinds of processes or two kinds of reactions taken place in the link reaction. Hence it is called as pyruvic acid oxidative decarboxylation, PAOD. So since pyruvic acid is an end product of glycolysis, right? So this end product of glycolysis is pyruvic acid. The pyruvic acid enters the link reaction, right? So pyruvic acid enters into the link reaction. After entering into the link reaction, pyruvic acid undergoes into different changes. The major changes are two. One is decarboxylation and the second one is oxidation. Okay, simple. Decarboxylation, whenever you hear the term D means removal. Okay, so removal of carbon. So removal of carbon in the form of carbon dioxide is called as decarboxylation. Okay, addition of carbon is carboxylation. Removal of carbon in the form of carbon dioxide is called as decarboxylation. Okay, then oxidation. So you might have heard several times, oxidation means addition of oxygen. Okay, addition of oxygen or removal of protons or electrons. So addition of oxygen or removal of protons or electrons is called as oxidation. Okay, so these two processes or these two steps are being taken place in the link reaction. Hence this particular link reaction can be called as pyruvic acid oxidative decarboxylation. Okay, so now for example, this is what the cell. This is the cell. Okay, so this entire portion is cytoplasm. This is what? Cytoplasm. For example, this is mitochondria. Just imagine it is mitochondria. So every mitochondria has two membranes. One is outer membrane and the second one is inner membrane. So this is cytosol, or you can say cytoplasm. So in cytoplasm, what takes place? Glycolytic pathway. So in glycolytic pathway, so glucose is the substrate. Glucose is a substrate which undergoes into 10 several reactions, and finally we have got two molecules of pyruvic acid. Okay, so two molecules of pyruvic acids are formed due to glycolytic pathway in the cytoplasm. Hence it is called as cytoplasmic respiration. Okay, now this pyruvic acid, this particular pyruvic acid contains 3 carbons. So this pyruvic acid enters into mitochondrial matrix. Okay, so it enters into mitochondrial matrix. Right, so every mitochondria is surrounded by two layers. One is outer layer and the second one is inner layer. Now what happens? This pyruvic acid is prohibited. This pyruvic acid movement is stopped by the mitochondrial membranes. Means mitochondrial membranes do not allow the pyruvic acid inside it. Okay, now what happens here? There are some channels, there are some proteins, there are some carriers on the mitochondrial membrane. For example, these are the proteins. What is this? These are the proteins which are called as, these proteins, they are a special type of proteins, they act as a carrier, or so these are the proteins which are called as pyruvate hydroxyl antiporter. Okay, so pyruvate hydroxyl antiporter, and the channel which allows the pyruvic acid into the mitochondrial matrix is called as what? This particular channel can be called as pyruvate channel or pyruvate translocator. Okay, so pyruvate channel or pyruvate translocator is a channel which allows pyruvic acid from cytoplasm to mitochondrial matrix. Clear? Now this pyruvic acid finally entered into the matrix of mitochondria. Now what happens after entering the pyruvic acid into the mitochondrial matrix? Clear? So now what happens? For example, this pyruvic acid undergoes into two steps or two reactions. One is decarboxylation and the second one is oxidation. Means pyruvic acid oxidative decarboxylation. Clear? So after pyruvic acid oxidative decarboxylation, generally pyruvic acid contains 3 carbons. Okay, so 3 carbon compound is lost one particular carbon in the form of carbon dioxide. Hence the number will be 2 carbons. That is acetyl CoA. Pyruvic acid becomes acetyl coenzyme A. This is 2 carbons compound. Okay, so this is a change, this is a modification which takes place in the matrix of mitochondria. Okay, so what are the steps that are being taken place during the conversion of pyruvic acid into acetyl CoA? We will see. Okay, so generally, so for the first, you can observe, so glucose is converted into pyruvic acid. Clear? So for example, I am writing here glucose, this is 6 carbon compounds, is converted into pyruvic acid, that is 3 carbon compound. 2 molecules or 2 moles of pyruvic acid is formed as an end product in the glycolytic pathway. Okay, so now pyruvic acid may be entering into next stage, that is link reaction, or you can say another next stage, that is Krebs cycle. Whenever the oxygen is available, I am quoting oxygen plus, whenever the oxygen is available, this particular pyruvic acid enters into Krebs cycle. Okay, if there is no availability of oxygen, for example, if there is no availability of oxygen, I am pointing out that is minus O2. If there is no availability of oxygen, this particular pyruvic acid enters into fermentation. Okay, so fermentation, enters into fermentation, right? So if pyruvic acid enters into Krebs cycle, link reaction, Krebs cycle, then followed by electron transport system, the final products will be carbon dioxide, water and some amount of energy. Okay, if this pyruvic acid enters into fermentation, the final output may be carbon dioxide is common. Along with carbon dioxide, some of the special chemical compounds like ethyl alcohol, lactic acid, etc. are formed. Okay, so this is about the path or fate of pyruvic acid. The fate of pyruvic acid is based on the availability of oxygen. If oxygen is available, this pyruvic acid enters into link reaction followed by Krebs cycle, then followed by ETS. Then if there is no availability of oxygen, pyruvic acid enters into fermentation process and the end products will be carbon dioxide and ethanol or methanol or lactic acid. Okay, so this is about the fate of pyruvic acid, or you can say pyruvate. Okay, now if the pyruvic acid enters into the matrix of mitochondria, this pyruvic acid loses one particular carbon and it becomes into acetyl-CoA, that is a 2-carbon compound. Nothing but what? Decarboxylation takes place in the formation of pyruvic acid into acetyl-CoA. Okay, so what kind of changes are being taken place during the conversion of pyruvic acid into acetyl-CoA? Let us discuss. For example, the broad equation I want to write on the board. Just have a look. 2 moles of, for example, you take 1 mole first. So pyruvic acid, Pa stands for pyruvic acid. This is 3-carbon compounds. Clear? Now this pyruvic acids lost 1 carbon in the form of carbon dioxide. So during this particular conversion, NAD plus nicotinamide adenine dinucleotides, NAD plus, can be converted into NADH2. Okay, nothing but addition of protons are taken place. That is called as what? Addition of protons from pyruvic acid. Nothing but pyruvic acid is the compound which lost the protons. These protons are added to the NAD and NAD becomes NADH2. Clear? So this is purely an oxidative mechanism. This is decarboxylation, this is oxidation. Means oxidation taken place and decarboxylation taken place during the conversion of this particular pyruvic acid into acetyl-CoA. Okay, so acetyl coenzyme A. So this is the 2-carbon compounds. Clear? So 3-carbon compounds, that is pyruvic acids, can be converted into 2-carbon compound, that is acetyl-CoA. Right? 1 carbon is lost in the form of carbon dioxide and 2 protons are lost from the pyruvic acid. These 2 protons are added to the NAD and NAD will become into NADH2. Okay, now there are some cofactors, there are some factors which are involved in the conversion of pyruvic acid into acetyl-CoA. So 5 kinds of factors. Remember, magnesium ions, Mg+2 factor is involved in the conversion. Then TPP, so TPP stands for thiamine pyrophosphate. So thiamine pyrophosphate is involved. Then NAD, already I have written here, NAD, nicotinamide adenine dinucleotide, is involved. Then LA, so LA stands for lipoic acids. What is that? Lipoic acids. Lipoic acid is also involved in this particular reaction. Then FAD, so FAD, CoA. Right? So CoA, coenzyme A, CoA is also involved in this particular conversion of pyruvic acid into acetyl-CoA. Clear? So these are the 5 factors. One is magnesium, thiamine pyrophosphate, nicotinamide adenine dinucleotide, lipoic acid and coenzyme A. So these are the 5 factors which are involved in the conversion of pyruvic acid into acetyl-CoA. What happens here? 3-carbon compound becomes into 2-carbon compound. Clear? So one carbon is lost in the form of carbon dioxide, that is decarboxylation, and the NAD will become into NADH2, that is called as oxidation. This entire conversion is catalyzed by a complex enzyme called Pyruvic Dehydrogenase. What is that? Pyruvic dehydrogenase is a complex enzyme which catalyzes the conversion of pyruvic acid into acetyl-CoA. Very important. So this complex nothing but there are several enzymes in this particular complex. So what are they? Number 1, Pyruvic decarboxylase. Pyruvic decarboxylase is an enzyme. Number 2, dihydrolipoyl transacetylase. So dihydrolipoyl transacetylase. Number 3, so dihydrolipoyl dehydrogenase. So these three are the enzymes in this particular complex enzyme, that is pyruvic dehydrogenase. Okay, pyruvic decarboxylase is one of the enzymes. Second one is dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. Clear? So these three enzymes are there in this particular complex enzyme. Now what happened? Just see what happens. So what kind of reactions will be seen in the conversion? Just see and observe. Right? In the first step, pyruvic acid combines with TPP, that is thiamine pyrophosphate, in the presence of, what is the first enzyme? That is pyruvic decarboxylase. So in the presence of pyruvic decarboxylase, this particular pyruvic acid that contains 3 carbons and the cofactor called TPP, thiamine pyrophosphate, these two compounds combines together and there is a formation of TPP complex. What is that? TPP complex, and there is a removal of carbon dioxide. Okay, removal of carbon dioxide can be called as decarboxylation. Means decarboxylation is occurred in this particular conversion. Number 1. Then TPP complex. TPP complex combines with La. La stands for, this is one of the factors of conversion. Lipoic acid. La stands for lipoic acid. So lipoic acid will lead to acetyl lipoic acid complex, La complex. Acetyl lipoic acid complex plus TPP is removed. This particular conversion is catalyzed by the second enzyme called dihydrolipoyl transacetylase. Okay, so dihydrolipoyl transacetylase is an enzyme which catalyzes this particular conversion. Remember, so La is in oxidized form here, oxidized form of La. Clear? Then acetyl lipoic acid complex, acetyl lipoic acid complex combines with coenzyme A, coenzyme A, in the presence of third enzyme, that is dihydrolipoyl dehydrogenase. Then finally we will get acetyl CoA, that is 2 carbon compound, plus lipoic acid, that is reduced form. So reduced form of lipoic acid is formed, and the final output of link reaction, that is acetyl CoA, that is a 2 carbon compound, is also formed. Then lipoic acid, that is reduced form of lipoic acid, combines with NAD plus. Then finally NADH2 is formed and lipoic acid oxidized form is also formed. So this is about different stages or different steps of link reaction. See, there are basically 1, 2, 3. So these 3 steps are catalyzed by 3 kinds of enzymes in the complex enzyme, that is pyruvic dehydrogenase. One is pyruvic decarboxylase, catalyzes the first reaction of link reaction. Second enzyme catalyzes the second reaction, and finally third enzyme catalyzes the third reaction of link reaction. Clear? Now just observe, what is the substrate of link reaction? Substrate nothing but the compound or substance which enters into the reaction. What enters into link reaction? Pyruvic acid. Okay, so pyruvic acid is a substrate for link reaction. Then what is the outcome or end product? Acetyl-CoA. So acetyl-CoA is an end product of link reaction. Pyruvic acid is a substrate of link reaction. Remember, now this particular acetyl-CoA, the fate of acetyl-CoA may be different. How the acetyl-CoA enters into different metabolic activities. Just see how the acetyl-CoA may be entering into different metabolic pathways. Right? So this is what acetyl-CoA. Acetyl-CoA is formed finally. So this acetyl-CoA may be entering into the next level of aerobic respiration, that is Krebs cycle. Okay? So acetyl-CoA may be entering into the Krebs cycle. That is the first option. Then the second one is acetyl-CoA may be come out from the matrix of mitochondria through malate aspartate shuttle. These are the carrier proteins. This is the channel, that is malate aspartate shuttle. Malate aspartate shuttle. Okay, so this channel allows the acetyl-CoA from matrix to cytoplasm. Okay, so where this acetyl-CoA involves in the beta oxidation of fats. Okay, so this is a fat metabolism. Beta oxidation of fats will be started by a substance called acetyl-CoA. Right? So acetyl-CoA has two options. It may be entering into the Krebs cycle in the matrix of mitochondria. It may be entering into the beta oxidation of fats, which will be taken place in the cytoplasm of the cell. So for that, this acetyl-CoA comes out through the particular channel called malate aspartate shuttle. Okay, so now we will see the third stage of aerobic respiration, that is Krebs cycle, in the next session or next class. So today, so this is about the entire picture of link reaction. Okay, so in next session we will see the Krebs cycle. Thank you one and all.